There was in 1983, just after I published the first book, a very interesting article that was published by two American scientists, Carl Ganz and Glenn Northcutt, who developed the idea in science that the neural crest was very important in the origin of vertebrates because this structure made it possible to develop what they called the new head of the chordates. That means the head of vertebrates. The head of vertebrates in which the brain has developed. And the brain has developed particularly from the lateral region of the neural plate to form the telencephalon, from which the brain of human, which is with its extraordinary complexity and has been produced by evolution. And I would like in the third part of this talk to show you that the work that we have been doing during the recent years is very much in favor of the very great importance of the neural crest in the evolution of vertebrate, and particularly in the evolution of the brain and the craniofacial structures. So we have looked at the molecular control of the development of the neural crest in these regions. And one thing that was extremely striking in the experiments that we did before and that I have just described is the fact that the neural crest, which is so important for the development of the brain because it provides vascularization, because it provides protection to the brain, and of the face, because it constructs all the skeleton of the face, is the OX negative domain. That means this region which is in front of rhombomer 3. So we wanted to explore a little more what is the role of these OX genes in development. And so this is just to remind you that the skeleton, that means the frontal, parietal, squamosal, optic capsule, upper beak, and lower jaw that you see here, and also is derived from the OX negative domain of the neural crest, that we have called it facial skeletogenic neural crest, or FSNC. By opposition to the posterior one, which gives rise to the heart structures, vascular structures, and also to the hyoid bone, at the exception of the antoglossum and part of the basial, and which is here, and which is the OX positive region. So what we wanted to know, since we are embryologists, and we know that there is a lot of capacity of regeneration in embryos early, we wanted to know whether this FSNC, facial skeletogenic neural crest, going from the posterior diencephalon down to rhombomer 3, is dispensable. In other words, whether if we remove it, it can be replaced by neural crest cells coming from the OX positive domain. You see that it forms these cells, these neural folds which express slug. So Sophie Creuset in the lab decided to remove these neural folds which express here the slug gene, and which goes from the mid-diencephalon, that means the beginning of expression, down, and she removed it down to the level of rhombomer 3. And you can see that in the control at 9 somite stage, you see the cells which have already migrated from this neural fold and which form these two regions which are expressing of cells which are expressing slug. But of course, in the embryo in which the FSNC has been ablated, there is absolutely no such cells which have migrated. You notice that the neural tube, however, is closed here in the absence of neural folds. What is going to be the fate of these embryos? They do not have the FSNC, but they have plenty of neural crest cells, just more caudale. Are these neural crest cells more caudale going to replace the anterior one? The answer is no. You see how these animals look at embryonic day 6? They do not have a face. And they have their brain, which is completely open. So there is no regeneration from this posterior region. However, if inside this FSNC which has been removed, là, here, we insert a little piece which is about a third of the FSNC domain coming from a quail embryo into this level, you see that the phenotype is completely rescued. Which means that this little group of cells, which has been implanted there, has replaced the whole domain. And not only it is rescued superficially, but you see that also the skeleton has developed. And if you look at this skeleton, you can see that it is made up of cells coming from this graft, because the graft is quail, so the cells forming this skeleton is all quail. This skeleton can be, this same result can be obtained if you implant any part of this FSNC, and if you put it in any region of this FSNC area which has been removed in the host. In contrast, if you graft the complete region or part of the region of the posterior region of the cephalic neural crest, which expresses OX genes, the OX positive domain, into this FSNC area, you see the phenotype of the embryo is exactly as if you had done nothing. That means there is no face and there is anencephaly. And if you look at the brains after FSNC ablation, you see that this is a dissection of a control brain at E6. You see that the forebrain, the telencephalon, diencephalon, telencephalon, diencephalon, mesencephalon, rhombencephalon. And you see what happens after removal of the FSNC. The brain is flat. And the neural tube was closed to begin with, but it opens again. And there is absolutely no development of this telencephalon, that means in the form of the forebrain. So the conclusion at that point is that the OX positive domain, from rhombomere 4 to rhombomere 8 of the cephalic neural crest, cannot replace the OX negative domain to generate a facial skeleton. The OX negative domain of the cephalic neural crest behaves like what we call an equivalence group. What does that mean? That means that any part of this domain can replace the whole. So any part is equivalent to the whole. So any part, all the parts are equivalent between them. And the consequence of this is that the patterning of the facial skeleton is not crestin built. So each part of the neural crest in this FSNC does not know if it has to make a squamosal or Meckel's cartilage or whatever. The neural crest can do that, but they have to be told by environmental cue what to do exactly. Now, in order to be sure that what we see when we remove this crest, and when we see that the OX positive crest cannot replace OX negative crest, is it because these cells express OX genes? In order to know that, we are going to force the expression of OX genes in cells which are naturally OX negative. And this was done also by Sophie Creuset, by electroporation. Alors, the electroporation is done not only with OXA2 cDNA, but also with cDNA of green fluorescent protein. So the two constructs are in the same solution, and this solution is put in the groove of the neural tube at the 5 or 6 somite stage, and you have the two electrodes which are placed on each side, negative on left and positive on the right. And the nucleic acid are going to the positive side. Therefore, on this embryo on the top, there will be only the right side which will be electroporated, the left side which remain intact. So, in order to have the two neural folds which will be electroporated, which will be transfected, you have to take another embryo and inverse the electrodes, which is done. And what is done after is that the neural folds are removed, and you have two different neural folds. The two ones which have been electroporated and the two opposite ones which have not been electroporated. The two which have been electroporated will be grafted in one embryo, which will be the experimental embryo, and the ones which have not been electroporated will be grafted in another embryo, which will constitute the control. And what happens? First, the day after this operation at E2, you can see in this embryo, which is observed in UV light, that the GFP, that means the neural crest cells, which have been electroporated, have expressed the GFP gene, because the neural crest cells have migrated, and the whole head of the embryo is fluorescent. If you look at expression of OXA2, this is on the left, you see the normal pattern of expression of OXA2, which goes up to the limit between Rombaumer 1 and Rombaumer 2, and which is not at all expressed in the head. But in the embryo in which OXA2 cDNA has been electroporated, then you see that there is expression in the neural crest which cover the head. The result. Look at the embryo here. This is a control. That means the side which was not electroporated, which gives rise to normal phenotype. But here it is not normal at all. There is no face, profile view, and there is anencephaly, which means that the phenotype of the phenotype of the embryo in which forced expression of OXA2 gene has been produced is the same as the phenotype that you get when you remove the FSNC. That means OX negative neural crest. Meaning that the absence of OX gene expression in the cephalic neural crest is really critical for the development of the facial skeleton and of the preotic brain. Moreover, expression of OXA2, as you have seen, but also of OXA3 and of OXB4 that we have done in another experiment, in the OX negative domain of the neural crest, prevents cartilage and bone differentiation from neural crest cells and generates severe malformations in the preotic brain. thus reproducing the phenotype of excision of the facial skeletogenic OX-negative neural crest. So the next step, in order to analyze what are the molecular causes of these malformations, was to do other experiments with the excision of the facial skeletogenic neural crest and looking at gene expression modifications which result from this in the head of the embryo. And the first genes that we have looked at is FGF8, because FGF8 has been shown to be playing a critical role in development of the skeleton and also of the brain. And what we see after excision of this facial skeletogenic neural crest is very striking. You have on the left the normal pattern of expression at 22 somite stage in the embryo, which is normal. There are 3 pods which are very important of expression. The first one is the anterior one. The anterior one, là. See here, which is called the anterior neural ridge, or ANR. The second one is the isthmus, that you can see dorsally. This is the region between the midbrain and the hindbrain. And there is another spot which is very important. This is here in the branchial pouches, the branchial arches. If you look at the expressions in these different spots in the embryo in which you have removed the FSNC, you see first that there is no development of the forebrain. You can compare. And that the ANR is not completely abolished as far as FGF8 expression is concerned. There is a little spot which remains. In the isthmus, there is no much change, and there is absolutely no expression in the branchial arches. So we saw that what was very striking was the loss of expression of FGF8 after this operation. So we tried to rescue the phenotype by providing the embryo with exogenous source of FGF8. And for that, we took recombinant FGF8 and put it in Cefadex beads, which were soaked with FGF8. And the idea consisted in removing the FSNC, as you can see on the left, and putting the beads, which are red, on the lateral sides, which correspond to the ectoderm of the future branchial arch 1. And in this situation, you see in the upper picture the embryo when there is no extra source of FGF8 with anencephaly and no face, and you see the rescue of the phenotype here with the brain which develops better and the face which the facial buds which grow, and you see even that there is skeletal structures which arise. Therefore, the deficit is rescued at least partly by an extra source of FGF8. But we would like to know where neural crest cells which form this facial skeleton and which filled up the different buds, frontal buds, maxillary bud, mandibular bud, come from. And we look at the migration of the neural crest cells by using HNK1 in a normal embryo on the left, upper left, in the embryo in which there has been a complete removal of FSNC here, and in an embryo in which FSNC has been removed, but FGF8 beads have been placed laterally. You see the neural crest cells. This is this brown zone. This is normal. The neural crest cells with HNK1, you see that they have filled up branchial arch 1, maxillary bud around the eye, and also in on top of the telencephalon. But you see that there is none, absolutely none, when you have removed FSNC. They start here, but here it is not the head. But when you have put these beads, you see that there are neural crest cells and that they have invaded the first branchial arch. Where do they come from? We thought that perhaps they could come from R3, because R3 was left inside too, and R3 normally, as I said, provides a few cells to branchial arch 1. But it did not provide many cells. And it has been shown that R2 was preventing R3 to produce many cells. It was shown by the lab of Andrew Lumsden. So in the absence of the FSNC on the rostral part of the embryo, perhaps R3 could, if it could be stimulated by FGF8, provide enough cells to regenerate this facial skeleton. This is what we are going to look at. This is an experiment which was done by Sophie Creuset, a very nicely subtle type of experiment. In this embryo, you see the FSNC has been removed. You see on the left side that it is in yellow. And in addition to that, R3 of a quail, which is left, has replaced the R3 of the cheek on the right. The beads of FGF8 have been provided. If the cells come from R3, all the skeletons that we have seen, which develops in the rescued embryo, will be made up of quail cells. This is exactly what happens. You see the skeletal tissues, and you see that after, they are blue because the Alcyon blue labels cartilage, but you see the nuclei of the cells, cartilage cells, and these nuclei are QCPN positive, meaning that they come from the quail. Therefore, these quail cells from R3 colonize branchial arch 1 and colonize also the superior regions because the brain is closed. But R3 provides cells which primarily express OXA2 and lose expression when they reach BA1. But in this case, there are many cells coming from R3. So are they going to lose their expression of OXA2? And this experiment shows that it is yes. In the top, you see on the left that branchial arch 1 is filled up by cells which are QCPN positive, meaning that they come from R3, which is quail, in this same experiment as I described. And on the right, the next section, adjacent section, was hybridized with OXA2 probe. And you see that at that time, that means at E2.5, 1 day after the experiment, they express OXA2, as well as the cells which are in BA2 and in BA3. But if you look at the same experimental embryos, type of embryo, at E3.5, that means 1 day after, the neural crest cells which are still abundant in BA1 and which express the QCPN antigen, this is quail cells, do not anymore express OXA2. They have lost expression. This is why they are able to make this cartilage and bones. Now, we have found also that the beads can be placed in a different region, and they can be placed close to the ANR. That means more anteriorly. And in this situation, you see the 3 types of situation. The normal situation with the migration of the neural crest cells on the left. In the middle, no neural crescents because the FSNC has been removed. The R3 is still there. And in the right, the R3 is still there, and the beads of FGF8 are placed in the ANR, and the migration of the neural crest coming from R3 is more clearly attracted in the anterior region and covers the brain. You see, therefore, FGF8 exerts two effects on these neural classes. It stimulates considerably the proliferation of the cells and also acts as a chemoattractant, because you see in this experiment that the direction of the migration depends on the place where you put your FGFs. The next step was to see what is the effect of FSNC ablation on morphogenesis and gene expression in the developing brain, in the absence and in presence of exogenous source of FGF8. And this is just to see for morphogenesis that the FGF8 supply on the ANR restores brain development. You see the normal brain on the left. You see how the brain is after FSNC ablation. That means no development of the forebrain, or very little development of the forebrain, and the brain remains completely open. But when this extra source of FGF8 is provided, you see that it is completely different, and that the brain vesicles, the different brain parts of the brain develop. Now, what we have looked after that is a certain number, the expression of a certain number of genes which are critical for the development of the different parts of the brain. And you have here genes which are expressed on the dorsal midline, and particularly of the Wnt gene family. So Wnt1, as you see in the normal brain, is expressed dorsally in the midline. And of course, when the brain is completely open, there is nearly no expression of this gene except for a little spot on at the level of the posterior midbrain. But when the FSNC has been removed and FGF8 beads have been provided, you see that expression is restored nearly normally, together with the morphology of the brain. And for Wnt8b, you see also that the pattern of expression is restored when this FGF8 has been provided after FSNC ablation. Here you have an expression of genes which are critical for the development of different parts of the brain, and those are EMX2 and ELX2. And you see that it is very striking to see that the difference between the normal expression on the left, the very reduced expression on the brain, which has not developed normally after FSNC ablation, and the nearly complete restoration, which is apparent just by addition, after addition of FGF8. And this is the same for the OTX gene. And here what you see is that this is an expression of a gene which is sonic hedgehog, which is normally expressed in the basal region of the brain, and of course in the floor plate, and anteriorly also you see in the forebrain. After excision of the FSNC, the expression of a sonic hedgehog is expanded laterally, so that nearly the complete brain which remains after excision of the neural crest is expressing sonic hedgehog, as if the remaining of the brain is ventralized. And the addition of FGF8 restores nearly completely the development, the expression pattern of this gene with, as you see, it becomes very similar to, with the zonal emittance, and becomes very similar to that of the normal brain. What we have tried to know in the next step was whether FGF8 by itself could be able to do all these transformations that we see after FSNC ablation if the supply of neural crest cells coming from R3 were lacking. And in this, to answer this question, we have made excision of FSNC as usual, and in the panel which is on the right, we have removed R3. In this case, you see that the brain remains open, and that FGF8 supply by itself cannot rescue the brain morphogenesis. So it can rescue the brain morphogenesis by providing a growth factor to the neural crest cells of R3, and in this case, both together, FGF8 supply plus the neural crest can rescue the phenotype, but the neural crest is absolutely important for doing that. This is just to show you what happens to RhomboMER4 in a normal situation. Here it has been replaced by a quail, RhomboMER4 in this embryo, and you see that the cells coming from RhomboMER4 migrate to branchial arch 2. But in this case, in the absence of the FSNC and in the absence of RhomboMER3, but in presence of FGF beads, there is a certain stimulation of the growth of cells from RhomboMER4. But this stimulation is not very important, and the cells do not migrate to the head and do not reach the branchial arches, branchial arch 1. And if it could reach branchial arch 1, probably it would not regenerate the skeleton because it still would express the OX genes. So the conclusion of this series of experiments show that the development of the face, the facial skeleton, and the development of the brain are very interdependent, and that the link between these two processes is the neural crest. The neural crest is extremely important for making the ANR produce FGF8, because in absence of neural crest, the production of FGF8 in ANR is strongly reduced. And as a reverse action, FGF8, which is in the ANR, exerts a very important action on the growth and also on the migration of the neural crest cells. And these neural crest cells through FGF8 and by themselves also are absolutely necessary for the development of the telencephalon, and also on the limitation of expression of sonic hedgehog to the basal plate and to the ventral region of the brain. On the other hand, the neural crest increase and allows the development of the alar plate and increase the development of the alar plate and also of the roof plate of the forebrain and midbrain. Therefore, one can say that the neural crest plays a critical role in the processes, in all these processes. That means the development of the face and brain, which are interdependent. And it is essential for the development of the alar plate of the cephalic neuroepithelium. So I would like to acknowledge the collaboration of the people who have worked with me on these problems over the years, particularly Marie Métayer, Christiane Ayer-Le-Lièvre, and Gérard Couly, who were with me for many years, and now to José Brito, and particularly to Sophie Creuset, who has done all the experiments that I have described in the last part of this work. Thank you.